Section 1.1, what is science? So what are the goals of science? Science has three basic goals. One is to make explanations for events that happen in the natural world. And when we say natural world, we're talking about things on the earth, in space, in the Earth's atmosphere. We're not talking about things like maybe with ghosts, with supernatural things that cannot be explained, okay, or that can't be tested. They use those explanations to understand patterns in nature. For instance, in the fall, the leaves will come off the tree. They fall off the trees, and in the spring, the leaves come back out on the trees. So they try to understand what the patterns are in nature. And from that, they're able to make predictions. That's how they're able to predict the weather. They might be able to predict how tall you are going to be. They might be able to predict which strain of the flu is going to occur this year, and therefore they know what vaccine to make. Biology is not just a collection of never-changing facts. In fact, it is just the opposite. It's open to testing, discussion, and revision. So things in biology can still change, especially with the advent of new technology. For instance, they used to think that proteins were the genetic material, and now we know that DNA is the genetic material. Science is an organized way of gathering and analyzing evidence. So it's an organized way. It's not just haphazardly done. It's usually well planned out. And when they go to investigate something, it's not just, oh, let's see what's going to happen here. It's more trying to be a little more precise so that your results are valid. So what do scientists do? Well, they collect and then they organize this information, and from this they try to make an explanation. And they use these things that they've collected, the data that they've collected and organized, as evidence. Now, they may also test those explanations, see if it happens again, see if it happens differently, things like that. So as we said earlier, what were the three goals of science? Explanations, right? Natural explanations, patterns, and then to make useful predictions from those patterns. Now, science is also improving, and I told you that due to technology, it's always going to improve and advance. One thing that science does not do is prove absolutely anything. Okay, it does give an explanation for things and helps people to understand them, but it's not necessarily proven. It just may support a hypothesis. What procedures are at the core of scientific? So these are some of the things you should have remembered about observations, which may give you questions. You may make an observation that could give you an inference, and with this inference you would form a hypothesis. You can conduct controlled experiments. From the experiment you collect and analyze your data, draw conclusions, and then communicate the results. Think about an example of when scientific methodology could be used. The answer or the example I gave in class was maybe choosing clothes, what clothes you're going to wear. So there's a couple of observations you need to make, like what's the temperature going to be, what's the weather going to be like, what clothes do you have, what clothes are in your closet, depending on where you're going, what's the dress code there. And then you wear your outfit, and your data you collect could be people's reactions, whether or not you feel comfortable. And then from that data, you can decide whether or not to wear this outfit again next time, or maybe not to wear that outfit again. So when a question happens, you may be given a question, or you might have. As far as the inferring and forming a hypothesis, you might make observations, and then you get from your inferences, you may interpret your inferences, and from this inference that leads you to a hypothesis. So, the ground, say you come out of the movie theater, the ground is wet, you might make an inference that, oh, it must have rained, the water sprinklers may have been on. You can make a couple different inferences there, and that can lead you to a hypothesis. So an example of the hypothesis could be, if it rains outside, then the ground is wet. Now, when you design your controlled experiments, you need to test your hypothesis. This experiment has to test the hypothesis you are stating. And to do that, you have to control the variables and make sure that only one variable is changed. So why do you need to control variables? To make sure that you're testing your hypothesis. Now, when we're looking at variables, you always have what's called the independent variable and the dependent variable. Sometimes students have a hard time picking these out. So the dependent variable is the one that is deliberately changed. Deliberately. So you change it. And it is plotted on the x-axis. So I should be able to give you a graph like this one, and you should be able to tell which one's the independent variable. And in this case, it would be the heating time in minutes. This one here, what would your control group be? It'd be the group that did not get any heat, so it's not exposed to the independent variable, which is the heating time. Now remember this, a control group is used for comparison. That's what you're going to compare your experimental group to, is your control group. Now when we're looking at data, there are two types. You have what's called quantitative data and qualitative data. Quantitative are the ones that are numbers. You usually get this data by measuring or by counting. And qualitative data is a description. So it might say red. It might say rough. It might say smooth. Those are qualities. Okay, so that's your qualitative data. And there's no units on those. There'll be units with the quantitative data. So some of the tools that scientists might use include lab equipment. It's a picture of a beaker and a Erlenmeyer flask. Charts, graphs. And what's the difference between a chart and a graph? A chart is the one, or a table will be the one that has the numbers and the data on it. And the graph will be a picture representation of the charts. So here's an example of a graph. It shows how grass height has changed over time. What's the independent and dependent variable? So your independent variable is on the x-axis, so you should be able to tell me that. And then which one's the dependent variable? Good. Sometimes you may have errors, and that might change your data. Anytime, say we do an experiment in class and we have six groups, and 5 groups get almost the same exact data, and the 6th group doesn't. Usually it's because maybe they did a different procedure. Maybe they didn't do the experiment exactly the way it was written. So what you do, that's why we do 6 groups, as opposed to just doing 1 group and saying, oh, okay, this is the answer, or this is the correct data. Or we might do it in pairs. So in their case, we'd have 12 different groups of data. So the larger the sample size, the more reliable the data is that I have. This is plant growth in soils. This is what would the conclusion be? So you look at the pH, okay, and then the growth. So which pH grows, do the plants grow best in? Yeah, right around between 6 and 7, huh? Because 6.2, it jumps at 6.4 to 50 centimeters. Then it stays right around there between 6.4 and 6.8. Now you may have to do what's called revising your hypothesis. So say your hypothesis is not supported. Well, you got to go back and look at your observations again, give a different experiment, or maybe change your experiment, and then you'd have one example would be if you are looking at animals, behavior of animals, and then is a big one. You might until your hypothesis you don't do an experiment, you just kind of observe them and see how they interact. Sometimes ethics will prevent your experiment, so you may not always have a control group, especially if you're dealing with. So say you have a chemical that you think causes cancer. Well, you can't give the chemical to people because you really don't want more people to get cancer. So what you do is you look for volunteers who have already naturally been exposed to the cancer agent or the chemical that you think causes cancer, and then just compare them to people who have not been exposed to the chemical. But you still want to try to control as many variables as possible, and you want to study large groups of subjects. Why do you want to study large groups? Okay, because sometimes you might have individual genetic differences, and those might change or skew your results. So say you've got a group of family that cancer doesn't run in their family. Maybe they have a where they can block cancer, or they're immune to cancer. I'm just saying, say they have that. Well, then your data is going to be different if you have this group of people who are not predisposed to getting cancer.